Hi, my name is Hana Itani, an assistant professor at the American University of Beirut. Most of you know me as the chair of the Trainee Advocacy Committee. So every year at the Council of Hypertension Scientific Session, the Council honors in the Early Career Awards session, which is a highlight of the meeting for the trainees, four top scored abstracts, including two basic and two clinical abstracts that compete for the Early Career Award. Congratulations to all the finalists that were selected and for their outstanding research. If you haven't seen these talks yet, they are a few clicks away and you can find them under the on-demand sessions. We are excited to announce the winner of the Early Career Award, Chudan. Zhu presented a talk in addition to the finalists titled A New Gut Microbial Metabolite Derived from Low Fiber Intake. On the behalf of the Hypertension Council, TAC, and Early Career Committee, I would like to congratulate you for being selected as the winner of the Early Career Award. And as I mentioned, this is very highly competitive, and congratulations to you and your research group. Can you tell us where you are in your training?
So I've just started my PhD at Monash University. And what I presented was my honors project in my honors year, which in Australia is the fourth year of the undergraduate.
That's great. So can you tell us how you got interested in this research project?
So I'll jump in as Chudan's supervisor. First of all, I'd just like to say how proud I am of her for winning this award. This is her first conference ever. And I think this is quite amazing to see all the effort that she put into her honors year and what a beautiful start of her PhD. So in our lab, we're interested in dietary fiber and how that interacts with the gut microbiota and is able to lower blood pressure. And we and now others have shown that with high fiber intake, there is an increase in certain metabolites that are able to lower blood pressure. These metabolites are called, for example, short chain fatty acids. However, we know that there will be many other metabolites that are also produced by the gut microbiota that we still don't know what their role is. So in this study, we used an on-target metabolomic approach to try to discover what these other metabolites were, in particular metabolites that were produced by a low fiber intake. Because this is really important because we know that low fiber intake is associated with higher blood pressure. But then Chudan went on to find these amazing other metabolites that had potentially a role in blood pressure regulation. So we're really excited about these findings. Chudan, would you like to explain what you found?
So we discovered a biological pathway and a metabolite known as p-cresylglucuronide PCG, which the precursor is derived from the gut microbiota. So these two were adversely associated with with hypertension in both animal and human studies. And with a high fiber diet, the level of PCG was reduced in the animal study. And interestingly, our preliminary data showed that this metabolite PCG might induce inflammation as well. So in summary, our data suggests that this metabolite PCG might elicit inflammation in association with hypertension. And with a high fiber diet, its production might be mitigated in the body.
Thank you. These are great results that we've seen in the presentations. Can you describe a bit the experimental models that you've used, both in rodents and in the human studies that you've done? And can you tell us more about the translational aspect of your research?
So in this study, we used angiotensin II to mimic hypertension in mice. And we also used sham to as our control group. So in this study, we had two major translation aspects. So we had a combination of human and animal samples to show similar findings. And lastly, we also had an in vitro experiment which involves human PBMCs. Yeah.
Yeah, so what is important about this model in particular is that angiotensin 2 was not the major driver of the changes. The major driver was the diet. And we believe that that would be independent of the angiotensin or perhaps any other model of hypertension. There was a major difference in terms of blood pressure when animals are exposed to a lower high fiber diet. And this is driven, this change in blood pressure because of the diets is driven by the gut microbiota. We published these findings. They were quite important findings because of their role of low fiber intake in driving hypertension and also heart failure, cardiac hypertrophy and fibrosis. Those findings were published in Circulation this year. And what we're analyzing this time now is the effect of the fiber intake per se. But as Chudan mentioned, what is really important is the translational aspect, where we had 70 human samples that had ambulatory blood pressure measurement. And we're able to then determine the ones that had mass hypertension, white coat, and so on, to have like a true diagnosis of hypertension. And we showed that the same pathways is still associated with high blood pressure. And also, of course, importantly, also the in vitro studies that we use in human cells. So there are many translational aspects.
Thank you so much. These are great findings, as you mentioned, that you know, non-pharmacologically, we can alter diet and that will improve hypertension. And we can also, by identifying specific gut metabolites, we can probably interfere in treating hypertension. Thank you so much, and congratulations again on your achievement. And for the audience, if you haven't attended the early career sessions yet, you can click on the on-demand moderated sessions and you will find them there.
Thank you.
Congratulations.